Hello everyone, welcome to Bit of Anatomy. In this tutorial, we will see the features of the cleavage division. In the previous tutorial of embryology, we saw the process of fertilization. If you have not seen that video, do click the link that is appearing on your screen now. After the process of fertilization is completed, the zygote is formed, which is a single-celled entity. So that will undergo cleavage division to form multiple cells, and that cleaved embryo it will implant into the uterus. All these changes it occurs in the first week of the development. So if you see the fertilization process, it occurs within the ampulla of the uterine tube, and the cleavage division will begin as the embryo is passing from the ampulla of the uterine tube towards the uterus. So the entire cleavage division it occurs within the uterine tube and also partly within the uterus. So finally that cleaved embryo it will implant into the uterus. In this tutorial we will see the features of the cleavage division. So the entire cleavage division it occurs within the protective covering of the zona pellucida. So there is no true one cell stage. Immediately after the fertilization, the cell or the resultant zygote it will divide immediately to form two cells of unequal size, a larger and a small. There will be two polar bodies which will eventually disintegrate. So during the cleavage division, the large cell will divide first and then the small cell will divide. So with the division of the large cell, we can appreciate a 3-cell stage of the embryo. So next the small cell will divide, resulting in 4-cell stage of the embryo. So eventually the 4-cell stage is converted into 8-cell stage, and the 8-cell stage is converted into 16-cell stage. This 16-cell stage it appears like a mulberry, so hence it is also called as stage of morula. So in this morula, the cells are differentiated into an inner cell mass and outer cell mass. The uterine milk coming from the uterine glands, it will seep into the developing embryo and it occupies the space between the inner cell mass. So these smaller spaces it will coalesce and it gradually increases in size, or the space it will increase. So with the increase in the space, so the cells gets rearranged into an outer layer which is formed by the outer cell mass and an inner cell mass, and we can see a larger cavity called as the blastocyst. So with the appearance of this larger cavity, that is the blastocyst, we call this stage of the embryo as the blastocyst, which completes the cleavage division. So the cells are now differentiated into an outer cell mass which forms the trophoblast and which gives rise to supporting tissues of the developing embryo, and the few cells that are present in the inner cell mass, only these cells will form or will develop into the embryo, hence they are called as embryoblast cells. The part where the embryoblast is present, it is called as embryonic pole. The opposite pole is called as subembryonic pole. The trophoblast cells which cover the embryonic pole, they are called as polar trophoblast, and the opposite part is called as mural trophoblast. So these are the various stages that occurs in the cleavage division. So if you see the timeline, there is zygote, the 2-cell stage, the 4-cell, the 12-cell, the 16-cell stage. So zygote is formed on day 1, that is on the day of the fertilization. Then 2-cell at 30 hours, 4-cell at 40 to 50 hours, 12-cell at 72 hours, and 16 cells at 96 hours. So this is the timeline of the entire cleavage division. So the cleavage division, it converts a single cell zygote into a blastocyst, in which there are differentiation of the cells into an embryoblast and also the trophoblast layer. So do subscribe for future updates. Thank you.